Hello, I'm Dr. Trace Jordan and I teach at New York University. In this video, we're going to be investigating the virus that causes COVID-19. This module is the second part of a series of online educational resources about COVID vaccination. Here's the outline for the video presentation. We'll begin by examining the results of a COVID-19 test. Then we're going to ask ourselves a few questions. First, what is a coronavirus? What is the structure of a coronavirus? How does a coronavirus infect cells? How does a COVID-19 test work? And then at the end, we'll go full circle and revisit the results of the COVID-19 test. So let's begin with looking at a COVID-19 test. These tests are becoming increasingly common. I've had several tests myself. You may have had a COVID test yourself. So what is the type of feedback that you get from a COVID-19 test? So here's an example of a communication that was sent to John with information about his COVID-19 test. So the text of the email says, "Hi, John, your COVID-19 test is complete. Your results indicate that you're infected with the virus responsible for COVID-19. So that leads us to ask the question, what is the virus responsible for COVID-19? You may have heard that COVID-19 is caused by a coronavirus, but what exactly is a coronavirus and where does that name come from? That's what we're going to be looking at in this next section of the video. The image on the right shows a coronavirus. So before we go on, we can just pause and take a look at it. It has a yellow central region and green spikes that are protruding from the surface. In fact, a coronavirus is named for its appearance when viewed with an electron microscope. Viruses are very, very small. The diameter of a coronavirus is about 1,000 times smaller than the width of a human hair. In order to see a virus, we need a very powerful and sophisticated microscope called an electron microscope. These microscopes are capable of achieving magnifications of up to 100,000 times, which enable us to see viruses. The term corona means crown. And you're familiar with this term if you've ever heard about the coronation of a king or queen. And as we saw in the image, a coronavirus has spikes protruding from its surface that makes it look like a crown. So in effect, the term coronavirus means crown virus because of how the virus looks when viewed under an electron microscope. Coronaviruses belong to a family of viruses that contain many hundreds of different types. However, we're primarily concerned with 3 coronaviruses that cause disease in humans. And these coronaviruses cause respiratory diseases because they infect the upper respiratory tract in humans, which include the nose, mouth, and throat. The first coronavirus disease that was identified was called SARS. That stands for Severe Acute Respiratory Syndrome. The disease of SARS was first observed in 2002. In 2003, it expanded globally and was the first example of a pandemic caused by a coronavirus. A second coronavirus disease is called MERS, Middle East Respiratory Syndrome. That was first identified in 2012. Most recently, a coronavirus disease was discovered and has since caused a global pandemic. That disease is called COVID-19. COVID stands for coronavirus disease. 19 is used because the disease was first detected at the very end of 2019. At this point, it's important to distinguish between a disease and the causative agent of the disease. For example, COVID-19 is a disease. It's a set of medical symptoms that would be recorded by a doctor in a hospital. A microorganism that causes a disease is called a pathogen. Patho refers to disease and gen refers to origin. So a pathogen is responsible for the origin of the disease. The pathogen that causes COVID-19 is a virus called SARS-CoV-2. Full name of this virus is Severe Acute Respiratory Syndrome Coronavirus 2. The virus was given this name because of its close genetic similarity with the virus that caused the original SARS outbreak in 2002. Now let's ask ourselves, what is the structure of a coronavirus? What does it look like? What are its components? The diagram on the left shows a schematic view of a coronavirus. Essentially, it looks like a ball with blue spikes protruding from it. And that is something that we can keep in mind as we go into the details of the various coronavirus components. On the right-hand side is a cutaway view, a slice through the middle of the coronavirus that illustrates its various components. The genetic information of a coronavirus, including SARS-CoV-2, is RNA, ribonucleic acid. That differs from the genetic information in human cells, which is composed of DNA, or deoxyribonucleic acid. The packaging of RNA within the virus is assisted by a nucleocapsid protein, which is indicated by the letter N. Now, SARS-CoV-2 is classified as an enveloped virus. And the reason is that the viral genome is enclosed by what is called a viral envelope, which is composed of a lipid bilayer with embedded proteins. And the diagram illustrates some of those proteins. You see an envelope protein E, a membrane protein M, and most distinctively, the spike protein S. So the blue spike protein, which is labeled on the right, is represented in the diagram on the left as the spikes protruding from the virus. And the spike is particularly important for understanding the structure and function of SARS-CoV-2. So let's take a closer look at this very prominent spike protein. So in this diagram, you see that the spike protein has been enlarged, and we're getting a molecular level view of the protein in the box on the right. The spike protein is a trimer consisting of 3 amino acid chains that are intertwined with each other. In the diagram, these chains are colored green, purple, and cyan. So they're long chains of amino acids that constitute the spike protein. The virus uses its spike protein to attach to human cells. So it is essential for SARS-CoV replication and infection to utilize its spike protein. In addition, the spike protein also generates a human immune response. And this immune response to the spike protein has been utilized in the development of several types of vaccines, which you will learn about in other modules in this series. As we saw earlier, SARS-CoV-2 contains an RNA genome. In fact, the RNA genome of coronaviruses are the largest of any type of RNA virus. The SARS-CoV genome contains almost 30,000 RNA bases. However, that's still much, much smaller than the genome that we have in human cells, which consists of 3 billion DNA base pairs. The genome is organized into regions or sequences of DNA bases that encode information to make viral proteins. Some of those proteins are enzymes that assist with viral replication. So one example that you'll see in the middle of the slide is an enzyme called RNA-dependent RNA polymerase. So that is an enzyme that makes more copies of RNA, which enables the virus to replicate. The region of the genome shown in green on the right-hand side codes for what are called structural proteins, which help create the overall structure and composition of SARS-CoV-2. In particular, I'd like to point out the region of the genome that codes for the spike protein S, which we illustrated in previous slides. Like any virus, SARS-CoV-2 mutates and evolves, and that has important consequences for understanding its ability to infect cells, and also potentially the ability of vaccines to work on a long-term basis. Mutations cause changes in the RNA genome of SARS-CoV-2. For example, one base in RNA could be replaced by another. A base could be deleted. Other bases could be inserted. So there's a variety of different mutations that can occur in the RNA genome of the virus. One SARS-CoV-2 variant, which has attracted particular attention, has a single amino acid change in the spike protein. This variant is called D614G, and the reason it is given that designation is that 614 refers to the 614th amino acid in the amino acid chain within the protein, and the original amino acid D, which is shorthand for aspartic acid, is mutated into a different amino acid G, which is shorthand for glycine. And you see in the picture on the right, the location of this particular amino acid mutation, D614G. Viral variants, such as this one, are subject to evolution by natural selection. So as with any type of natural selection, a genetic variant that has a selective advantage will tend to reproduce and become more successful within the population. And we're starting to see evidence for that for this particular variant of SARS-CoV-2. Studies in the lab have shown that this particular variant D614G is more efficient at infecting cells, and also when it does infect cells, the virus reproduces in greater numbers. Also in human populations, we're starting to see an increase in the frequency of this particular viral variant, indicating that it is achieving a certain selective advantage in the population of viruses that are being tested during COVID screening. Let's now look at how a coronavirus infects cells. In order to infect a cell, SARS-CoV-2 has to find a point of attachment to the cell. And it does that by binding to a protein called the ACE2 receptor. ACE2 stands for angiotensin converting enzyme 2. This is an important enzyme in human health. It has a variety of functions, including regulating blood pressure. And what the virus does is it co-opts this enzyme and uses it as an entry point to attach to human cells. So the diagram here shows that the spike protein of SARS-CoV-2 binds to the ACE2 protein, which acts as the receptor. So that binding between the spike protein and the receptor is the first stage that is necessary for the virus to infect a cell. Once that binding has happened, the envelope of the virus is able to merge with the membrane of the infected cell and inject the genetic material into the cell. And so that is the first stage of viral replication. This diagram shows the various stages by which a SARS-CoV-2 virus replicates within a host cell. So the first stage, which we talked about, is that the spike protein binds to the ACE2 receptor that provides a point of attachment. Then the viral envelope fuses with the cell membrane to inject the viral components. At stage number 2, the virus releases its viral genome composed of RNA. Then what happens is that the RNA is used by the ribosome within the human cell to translate the genetic instructions in RNA into an enzyme called a viral polymerase. Viral polymerase is capable of making more copies of viral RNA, essentially amplifying and replicating the genetic instructions of the virus. In stage 4, there is RNA replication, which is making more copies of RNA, and also transcription, by which the RNA is used to make complementary copies of other types of RNA strands. This creates replicated genomic RNA and what are called subgenomic transcripts, which encode the genetic information to make proteins. The translation of viral proteins happens within the cell at the endoplasmic reticulum, also by ribosomes, which are protein-making factories. So the replicated copy of the genomic RNA plus the copies of the viral proteins combine with the nucleocapsid, which organizes the RNA, and you create the structure of a new viral particle, which is called a virion. So a virus particle is named a virion. And that virion, a virus particle, can then exit the cell by a mechanism called exocytosis. Exo meaning outside, and cytosis referring to the cell. So essentially just leaving the cell. Although there are a lot of steps in this viral replication cycle, the main take-home message is that SARS-CoV-2, like any virus, co-opts the molecular machinery of human cells to make more copies of itself. And it's very devious in the way that it does that. And so once a cell is infected by SARS-CoV-2, it unwittingly makes many, many more copies of the virus, which enables the virus to replicate and propagate within the body. At the beginning, we looked at the results of a COVID-19 test. So now that we have some knowledge about SARS-CoV-2, how can we relate that to understanding how a COVID-19 test works? So a diagnostic test for an active SARS-CoV infection detects various components of the virus. And I've highlighted those two components here in this diagram as a reminder of our discussion of the SARS-CoV structure in earlier slides. A PCR test detects regions of the RNA viral genome. So PCR is a technique called polymerase chain reaction. It was invented in the 1980s, so it's been around for a long time. And a PCR test is considered to be the gold standard test for detecting the presence of a SARS-CoV-2 infection. So PCR test detects the viral genome. An antigen test detects viral proteins. An antigen is shorthand for an antibody generating molecule. In essence, any molecule from a foreign invader, such as a virus, which initiates an immune response by the body. Antigen tests often use the nucleocapsid protein N. So it's important to make the distinction when we talk about a SARS-CoV test about what type of test are we performing. Are we performing a PCR test to look at regions of the RNA viral genome, or are we performing an antigen test to look at the presence of viral proteins? So let's go into each of these tests in a little bit more detail. So here's a diagram of a nasal swab, which is the first stage of a PCR test. I've had one of these done myself. So what exactly does a PCR test do? So PCR, polymerase chain reaction, is a technique that amplifies the quantity of DNA. However, the viral genome of SARS-CoV-2 is composed of RNA, not DNA. So how do we get around that mismatch? Well, that can be done by converting the viral RNA into viral DNA using a process called reverse transcription. So you probably learned in your biology courses that the process of transcription is when DNA is copied into a complementary version of messenger RNA. Reverse transcription is the reverse of that process, where the sequence of bases in RNA is copied into a complementary strand of DNA. So the diagram at the bottom shows the stages of a PCR test for SARS-CoV-2. So first of all, there's sample collection, usually by nasal swab. Then there's extraction of the RNA, which is the viral genome. Then there needs to be the process of reverse transcription, where the viral RNA is converted into DNA. And once we have the genetic information in the form of DNA, we can use polymerase chain reaction. Real-time PCR is a version of PCR which can monitor the amplification of the DNA in real time. So you can watch the number of copies of DNA rise. And what you do is you use fragments of DNA called primers, which can detect specific sequences within the viral DNA, which are unique for SARS-CoV-2. So it's a very sensitive technique which can identify the unique genetic signature of SARS-CoV-2. So this has been used since the beginning of the COVID-19 outbreak and is again considered to be the gold standard of COVID-19 testing. More recently, there has been the availability of antigen tests. As we learned, an antigen is a protein of the virus which stimulates an immune response. So here is a diagram of an antigen test. Sometimes they're called rapid antigen tests because they can be performed quickly, and they can also be performed at home. So antigen tests have the capacity for quicker diagnostic and also being done at home without needing to go to a clinic. So an antigen test detects a viral protein, in this particular case, the nucleocapsid protein. So this is a schematic diagram of how an antigen test works. What happens is that in here, the antigen, which is the viral protein shown in green, and before you apply the antigen to this strip, you layer down a series of antibodies, which are designed to be complementary and bind the antigen. So the test essentially uses a strip where selected antibodies bind the antigen, and that is the positive test. If the antigen is present, then there is a detector molecule that produces a line on the test. So that indicates that you have a positive antigen test. It's also important to have a control line as well to make sure that the test is working. So if you have a positive antigen test, what you will see is a line corresponding to the existence of the antigen and a second line corresponding to the existence or the presence of the control. If you have a negative antigen test, you will only see one line, which is the control line, which indicates that the test is working. So again, an antigen test is looking at viral proteins, whereas a PCR test is essentially detecting viral RNA. And they both have their merits as different types of testing methods. So are there other types of COVID-19 tests? And in fact, there are. And the one test that I want to talk about is called an antibody test. Now, that's a little different from the test that we've already talked about, because an antibody test is not detecting regions of SARS-CoV-2. It's actually detecting the body's response to the SARS-CoV-2 infection. So an antibody test detects human antibodies. So infection with SARS-CoV-2 generates an immune system response, which includes the production of antibodies to various proteins within the virus. And this antibody test detects the presence of these human antibodies. One thing that is important to bear in mind is that antibody production has a time lag and persists after infection. So whereas the PCR test and the antigen test looks at the presence of the virus in the body, the antibody test is looking at the body's response to the virus, which takes time. Different antibodies rise over a period of weeks in response to infection by SARS-CoV-2. This test is also called a serology test because it uses blood serum, and it can be used to see if someone has been exposed to the virus and whether there are antibodies in the blood serum that have been generated in response to that exposure. So now that we've learned about the structure of coronavirus and the different types of COVID-19 testing, let's return to the original COVID test result that we saw at the beginning of the video. So here it is again. Hi, John, your COVID-19 test is complete. Your results indicate that you're infected with the virus responsible for COVID-19. So now we can unpack that statement because we've learned a lot more about what goes behind these tests. So first of all, the virus responsible for COVID-19 is a type of coronavirus, in particular SARS-CoV-2. And we learned that this virus has an RNA genome and spike proteins that it uses to attach to cells during the process of infection. One thing that this does not say is what type of test was performed. So that would be something that we might want to follow up with and learn more about. And we learned that there are various types of tests for the virus, in particular the PCR test and the antigen test. So that is the conclusion of the material that I wanted to present in this video. I do want to acknowledge the sources that I used to prepare this material. And finally, I also want to mention that this video is part of a series called Online Vaccination Science Resources for COVID-19 Education. I hope this video has been valuable to you, and I hope that you will watch other videos in this series. Thank you very much for your time.